Vegetative desiccation tolerance is the ability of a plant's leaves and stems to dry out completely, but then come back to life with that same tissue. Another way to define vegetative desiccation tolerance is that the plant undergoes a complete arrest in its metabolism, but then that can be restored upon rewatering. Air-dried state for vegetative desiccation tolerance can be defined as a threshold of about minus 100 megapascals, which is the equivalent dryness of air at 50% relative humidity.
Plants that possess this trait are also called resurrection plants because of the ability to be revived or resurrect after the vegetative tissues have dried out.
Plants with vegetative desiccation tolerance or the resurrection trait are extremely rare in nature. Only about 0.2% of all vascular plants show the resurrection trait. Plants with the resurrection trait occur across a wide range of lineages within the plant kingdom. We have plants that are in the moss family, also plants in the club moss family, like this Selaginella species here. We have ferns, and we also have flowering plants or angiosperms. This is a Ramonda species, and this is a Haberleia species, and we're using all of these species within our research programs.
Desiccation tolerance of spores, pollen, and seeds played an essential role in the successful colonization of almost every terrestrial habitat by plants. Because these reproductive tissues are desiccation tolerant, this ensures that vascular plants can continue to reproduce and evolve even if their vegetative tissues are dried up. We hypothesize that millions of years ago, all early land plants evolved the vascular systems, which allowed for the efficient transport and storage of water. All early land plants lost vegetative desiccation tolerance and instead relegated this trait into their reproductive tissues. The exact way in which numerous resurrection plants seemingly re-evolved this trait remains unanswered.
Everybody knows that any organism that is subjected to extreme desiccation dies. In plants, there is a fascinating property that seeds can be desiccated, stored for months, and when you water them, the little embryo inside the seed comes back to life. So this is a property of tolerating desiccation. But it's not only present in seeds, but several plant species can also tolerate desiccation in vegetative tissues, namely leaves and stems. The aim of the project is to investigate whether the circuit that activates desiccation tolerance in vegetative tissues is derived from the same circuit that activates this process during seed formation. To identify these circuits, we will analyze how genes are expressed and which metabolites are produced when the two different tissues are dehydrated. The idea is to identify the circuits that control desiccation tolerance in seeds that is linked to a developmental process, and the circuits that regulate desiccation tolerance in vegetative tissues like leaves that are responsive to environmental factors, and identify whether these circuits are the same but linked to two different processes during seed formation on environmental factors.
We are conducting this detailed transcriptomic and metabolomic analysis within different vegetative and reproductive tissue of resurrection plants to determine if they share the common aspect of desiccation tolerance.
We will conduct detailed transcriptomic and metabolomic analyses in several different tissue types of vegetative and reproductive of different resurrection plant species. The idea being that we will compare the desiccation response across the different tissue types. We are employing computational comparisons of transcriptomic and metabolomic data sets from the plants that we study. From this data, we are obtaining insights into exactly how these plants are able to adapt to extreme dehydration while other plants cannot.
We'll be conducting these detailed transcriptomic and metabolomic analysis using techniques that probe the responses of individual cells. We want to elucidate if distinct cell type specific desiccation responses occur in these cells, and the approach is called single cell sequencing.
In this project, we will be using two of the specialized techniques. That will be MALDI-MSI. It is a high-resolution mass spectrometry, and this will help in identifying the metabolites present in the plant tissue cryosection. And this will help us to compare and identify and define different metabolites profiling in plant tissue in different evolutionary lineages. And then another technique will be special transcriptomics. Through this technique, we can know how genes are expressed in a specific location within the plant tissue, which will help in revealing the tissue development and cell-to-cell interaction.
We are applying bioinformatics tools to sift through lots of genetic data to try and identify the exact genes responsible for these plants' ability to recover from a dried state once rewatered.
Omics techniques such as transcriptomics and metabolomics analysis help to identify the biomarkers in desiccation tolerance in plants with a comprehensive profile of metabolites and gene expression. In this way, the key compounds, genes, and the pathway will be identified that will assist in the discovery of biomarkers and that will aid in the development of the drought-resistant crops.
The trait that allows resurrection plants to temporarily suspend their metabolism for weeks, months, or years when desiccated, and then to become reactivated upon rehydration could be adapted one day to crop plants to minimize productivity losses under drought conditions.
As part of this project, we will study the DNA sequences or genomes of several resurrection species. By finding the important genes and processes that these plants activate to survive extreme water loss, scientists can learn how to make other crops better at surviving drought.